Assalamualaikum warahmatullahi wabarakatuh, and hi everyone. I'm Fatimah. I'm going to present on thalamic probabilistic connectivity with cerebral cortex in spastic cerebral palsy. This is my presentation outline. Let me introduce you with some keywords of this research. Cerebral palsy is a neurological disorder caused by lesions in the immature brain. CP is the most common motor disability in childhood, but it is unclear whether the pathways between thalamus to motor cortices are disrupted in spastic cerebral palsy. We use diffusion MRI to look at the microstructures of white matter tracts in vivo, no need for postmortem. It can also generate a quantitative measurement in the form of connection probability index, which refers to white matter tracts integrity. The objective of this study is to investigate the structural connectivity between thalamus and motor areas of cerebral cortex and its correlation with gross motor function measures in spastic cerebral palsy patients. There are three specific objectives of this study. First is to describe the pattern of distribution between thalamus to primary motor area, supplementary motor area, and premotor area. Second is to determine the correlation between the connectivity with GMFM score. And the third is to postulate the thalamus according to its connectivity to motor cortices. Let's have a look at the methodology of this study. We use probabilistic tractography done on diffusion MRI data. Then a quantitative measurement called connection probability index was measured between thalamus to each motor cortex. Correlation was performed between the connectivity with GMFM score. Then the thalamus was further parcellated or segmented into areas that corresponded to its connection with specific motor cortex. Diffusion MRI data was processed and analyzed using a software called FIMRIP software library. For tractography, we have one seed area, which is thalamus, and 6 target regions, which are primary motor cortex, for areas of premotor cortex and supplementary motor area. Let's have a look at the results of this study. The pattern of thalamocortical connectivity in cerebral palsy was variable according to the patient's clinical presentation. These are the graphs for each CP patients. The blue graph refers to the left connectivity in the left hemisphere, and the orange graph refers to the right connectivity in the right hemisphere. For CP1 subject, the left connectivity was lower than the right for all targets. This corresponded to the diagnosis of right hemiparesis. Let's have a look at the results for second objective. There was no significant correlation between thalamocortical connectivity and gross motor function measures. For the third objective, the result was in the form of images of segmented thalamus. For healthy control, the cluster that has positive connection to primary motor cortex, which is the green area, is related to the lateral group of thalamic nuclei. For postulation of thalamus in cerebral palsy, a striking feature was that there was a presence of cluster that has positive connection to supplementary motor area, which is the red area here. That was none in healthy control. Moving on to the discussion part. Large variabilities of connection probability index within and between the cerebral palsy patients, because each lesion brain was unique according to their clinical presentation, but some findings were not consistent with the clinical characteristics because several neuronal processes has occurred post-brain injury that leads to a functional reorganization of the neurons in the brain. Hemiplegic patients in this study showed a connectivity pattern that corresponded with their clinical presentation. They have lower values of connection probability indices in one hemisphere that caused contralateral motor impairment. This could be related with the perinatal stroke that is common in unilateral CP. For the second objective, there was no correlation between the connectivity with GMFM score because it is related with the large variabilities in the connection probability index in CP patients due to the morphological changes as the compensatory mechanism in the brain injury. Our small sample size could also affect correlation analysis, and other factors could also influence the GMFM score, for example, other tracts like corticospinal tracts or exercise therapy. For postulation of thalamus in healthy control, cluster that has positive connection to primary motor cortex were associated with a lateral group nuclei because it contained main motor relay station, which are ventral anterior and ventral lateral nuclei. For postulation of thalamus in cerebral palsy, presence of cluster that has positive connection to supplementary motor area because it gives a great contribution for motor function as a compensatory mechanism in brain injury. Here comes the concept of neuroplasticity, whereby the brain undergo morphological and functional modifications post-injury. In conclusion, there were vast variabilities and unique neuronal connections in cerebral palsy corresponding to the patient's clinical presentation. There was also an evidence of neuroplasticity as a compensatory mechanism for the motor deficit in CP. These are the list of some of my references. With that, I thank you.